As you can see on the screen, this is the 6th recorded lecture this spring. This is the last lecture. We will end our look at the digestive system, and this is chapter 25. This also ends the information for lecture exam 1, which I think is on the 4th of February. I had sent around an email yesterday, so today is the 22nd of January. When I'm recording this, I think I sent along an email on the 21st telling you that on the 4th of February, we're going to meet in person for that lecture exam. So even though we may still not have met for lecture, we will count on meeting together on February 4th for the first lecture exam, and this is the last lecture on that. Making good progress. As you can see here at the start, I think there's some things that might be of interest beyond the simple basics of anatomy and physiology of the digestive tract. Some things that we're going to learn about today as we take a look at the intestines. So this will be both small and large, and we will come all the way to the end, to the anus and defecation. So the small intestine was a place where I had said already, most absorption of most nutrients occurs here. And so we'll look at a few things like how on earth do we break down and absorb all the nutrients that we get from our diet. Second, we'll talk a little bit about lactose intolerance, which for some of you might be an issue. And for others, like me, who enjoy our dairy products as much as possible, it makes us a little more empathetic for those who are not tolerant as an adult of lactose. What causes farts, essentially, flatulence, and finally, something that could be urgent in your life. You could suddenly feel a need to go to the bathroom and sit on the toilet and defecate, and so we'll actually talk about how that's all controlled. So let's take this a step at a time and start out with the small intestine. So our story left off looking at the liver, and the gallbladder, and the pancreas, and how each of those is contributing directly or indirectly to what's occurring in the small intestine, and specifically in the duodenum of the small intestine. So here on this chart, you see the duodenum is the first section, and then it identifies subsequent portions of the small intestine. The duodenum, jejunum, and ileum are the sequence of the 3 sections of the small intestine. Some overview here on the small intestine. 1, I've already said this, most chemical digestion and absorption occurs here in the small intestine. This is the longest portion of the digestive tract, and in a living person, so this is important to note here, in a living person it's about 5 meters long. But in a cadaver, you'll find it to be about 8 meters long. So in a cadaver, in a dead person, it gets longer. And you have to ask why. And that's interesting because when we look at cadavers or cat anatomy, for example, we're thinking that it's a good proxy, it's a good example of the living condition, and yet things change. Well, why is because there's no muscle tone. And so though the small intestine in real life has lots of contractions that shortens its length, once we die, those muscle contractions relax and the small intestine gets even longer. And by the way, the small intestine does not refer to the length, but instead its diameter. And it's more narrow in a diameter than the large intestine. So it's about 1 inch in diameter in a human. As you can see in this image of this artwork, and then we'll look at some cadaver images coming up here, it's a highly coiled tube folding back on itself over and over again. And as it does, down there in the lower abdominal region, it's filling most of that space. So if you get punched in the stomach, well, if you don't get actually punched in the stomach, it's proper. You get punched below it, closer to where your navel might be, your belly button. If somebody punches you there, it's largely being absorbed by this small intestine. And perhaps behind it, some of the force is being experienced by the large intestine, Punch low enough, could be urinary bladder. So duodenum, jejunum, and ileum, and they're different color-coded right here. Key functions. The duodenum is where most of the action's at in the small intestine. Begins, of course, at its junction with the stomach at the pyloric valve or pyloric sphincter. And slowly, so you remind yourself, I think we've covered this, the stomach does not empty completely suddenly in one dramatic surge. That isn't what happens. Instead, the contents of the stomach are slowly metered out, much like a waiting room in a doctor's office slowly empties as each person is attended to individually, so one at a time. So I often think of the analogy of our stomach as a waiting room and then this metering out. So if you've overeaten, you are not going to resolve your overeating problem quickly in one sudden absorption. Instead, what's going to happen is it'll happen a little bit at a time. Talked already about the fact that the pH in the stomach is very acidic, and as the chyme from the stomach enters the small intestine, buffers from the pancreas, we said, elevate the pH. We talked about bile acids already breaking up, physically breaking up fats. Pepsin that was activated in the stomach by the low pH is now inactivated in the small intestine. And all sorts of pancreatic enzymes, so enzymes released from the exocrine portion of the pancreas, are promoting then chemical digestion, a lot of chemical digestion. We are not going to distinguish between the functional roles of each section of the small intestine. Instead, I'm largely focusing on the duodenum, and then know that an awful lot of absorption occurs in the subsequent sections of the small intestine, and that is the jejunum and the ileum. So here's that highly coiled small intestine in a cadaver. And here is some artwork, 2525, helping you to appreciate some of the histology of what's actually going on. So most of the adaptations that we see in the walls of the duodenum are about absorption. Absorption first into the bloodstream, and then the transport of those nutrients to the liver and perhaps other places in the body. The histology, as we will see in lab, is a simple columnar epithelium. You might have looked at this in cell biology. It's a common place for us to show you a simple columnar epithelium. And perhaps you did some PAS staining that was bright pink of these mucus cells. And these mucus cells are called goblet cells because their shape kind of looks like a goblet, like you drink wine out of. So these goblet cells are sources of a lot of mucus that are coating and buffering this region right around the villi in the small intestine. The muscularis externa is got a thick circular and a thinner outer longitudinal layer. So when we look at the walls of the intestine, why is that? And the answer is because there's an awful lot of constriction that narrows the diameter, and that, as we'll see, is involved in churning the contents, churning the chyme with the various enzymes and fluids that are in the small intestine. The surface area of this highly absorptive surface, this highly absorptive surface actually is promoted by three adaptations. First of all, there are these circular folds called plicae circularis, which increase the surface area, you can see, by about 2 to 3. And they're not shown right here. Second of all, what is shown are these finger-like projections called villi. Now, many times I think students get confused and think that these are somehow projections of a cell. That is not the case. So villi have many cells on their surface. You can see that. They have an inner core of arteries, veins, and lymphatics that are coming up here. So here, lymphatic vessels is kind of greenish yellow. And this is for some fat absorption just as well. So blood coming into the villi by the arteries and then out by the veins, and also some fatty materials absorbed into the lymphatics. These villi, you can see, increased by a factor of 10. So for those of you that had calculus, just between the circular folds and the villi, you've increased the surface area by about 30 times. And then finally, there are on each individual intestinal cell, not the goblet cells, but these the simple columnar cells that are coating really the surface of villi. These each have microvilli on them, so those are per cell, and there may be 1,000 or more microvilli, and these easily increase the surface area by an additional 20 times. So that 2 to 3 times, let's go 3 times 10 is 30, times 20 is 600 times these various adaptations increasing the surface area in the small intestine. Good thing you started learning about surface to volume ratios in zoology. Here now we can see a little bit more of that histology. Here's that very thick muscularis externa, the circular muscles that we can see. Here's some of the submucosa glands, and then of course here's the villi that we're pointing out in both the artwork, and here's a scanning electron micrograph showing the surface. So if you cut open and looked at the epithelium of the small intestine, you'd see these finger-like villi sticking up. And then, of course, on those villi are individual cells with about 1,000 microvilli each. Here's one of these really beautiful slides, and essentially they highlight the villi of the duodenum. It's almost everything you see in the view. This is probably mucus or other materials in between the villi, but essentially everything you're seeing here are these finger-like projections called villi. These are now transmission electron micrographs of a few. It looks like 3. So this is the boundary right here between 1 epithelial cell, 2 epithelial cells, 3 epithelial cells. These are the microvilli at the surface. I had talked to you about a junctional complex between epithelial cells of the intestines, and that this junctional complex seals these cells together, just like a Ziploc seal on a Ziploc bag or whatever bag, wherever you're getting Ziplocs. And so materials should not be seeping from the lumen of the small intestine in between the cells. That it's not a passive absorption. Instead, it's an active absorption here at the epithelial surface, whereas you'll learn in probably biochemistry or molecular biology, there are specific chemical receptors sent to the surface that specifically are there and will be bringing in certain nutrients. Some things do move in by osmosis, and that's to be expected. Okay. So here, I'll go back. Here we can see on the right the microvilli, and then here one cell of the epithelium, the whole cell with its microvilli. So that's just one of the 3 cells in this image. Anybody got any idea what these things are? Looks like mitochondria. Yeah, because you can see an internal membrane over in here, and mitochondria have 2 membranes. I talked to you already about some churning that goes on. These circular muscles doing a lot of churning, and they're mixing then all the contents. So that's the chyme with all of the secretions. Helping to neutralize, of course, the contents quickly. Promotes better digestion, just like any mixing does, putting the ingredients together. The churning also allows then all parts of the chyme to come into contact with the mucosa. If it stayed as kind of a chunky chyme, you'd expect that outer part of the chyme in contact with the mucosa would be well absorbed, but maybe all the goodies that are in the central part of the lumen might not. So by churning and mixing, it's just like if you're cooking, guys. If you put eggs, so you got scrambled eggs, and you're going to be moving those scrambled eggs around so that all of the egg is touching the surface, the cooking surface at some point. And finally, some of the churning intestinal motility that's going on is going to move things along towards the large intestine. Now, here's something called segmentation that occurs in the small intestine, in which you can see essentially sub-compartments forming. These segments are a form of a mixing and churning, and it's not part of peristalsis. This is really about part of the mixing that we see. So segmentation in the small intestine is associated with a mixing. Here, in turn, is peristalsis. Peristalsis is more of a very long wave moving down the length of the intestine that's squeezing behind this chunk of chyme and moving it towards the large intestine. So by the time we get our way through the small intestine, the chyme has largely been absorbed. What can be absorbed is absorbed. And I want to tell you on these pages in your textbook, let me actually look this up real quick because I adjusted it on one of the slides and this may not be right. Let me see if it is still 974. Yes. So there's a section 25.6 that talks in detail about chemical digestion and absorption. So I'm going to go through this a little faster. There's really nice text paired with a spectacular table that helps to put this all together. A couple of really nice tables. All the way until you get to 2531, figure 2531 on page 980. That really is just a wonderful summary table. Okay. So here are the kind of things that are variously broken down and absorbed. These are macronutrients, and micronutrients. You will learn more about these kinds of nutrients and nutrition as you prepare. That is the next information for you to work on your own for lecture exam 2. Okay. Lecture exam 2. Let me look this up kind of quickly here. So when we finish with chapter 25, which is today, then we move on in the next lecture exam. I will lecture about the respiratory system, but you on your own will look at chapter 26, parts of chapter 26, and you'll study about nutrition and metabolism. So it'll really pick up on this right here. It's only, I think, 3 sections out of that whole chapter, so probably a little less than half of chapter 26 is on your own. And it's good to follow that up. So right after February 4th, you take that lecture exam. It'd be really nice for you to jump in and look at chapter 26. And it's largely this. It's the next logical step. Okay. So this figure 2531 that I'm about to show you, you're responsible for virtually everything in it. And it's a wonderful, wonderful summary figure. Now here on the screen, it's probably hard for you to see this. And that's why you want to look at page 980 in your textbook, figure 2531, and study all of these details. But if you look at the very top, I think you can make out at least this. Here it's showing carbohydrates that are in the chyme of the small intestine. And actually the chyme of the small intestine will be down in here. Here they first talk about some of the changes that occur in the mouth. And then finally anything in the stomach, and then finally small intestine. So that's carbohydrates. Next, proteins, mouth, stomach, small intestine, and finally fats, mouth, stomach, and small intestine as well. And then down here, when we're talking about carbohydrates and proteins, what's broken down both within the lumina, that's plural for lumen, the lumina of the stomach and the small intestine, and sometimes is absorbed or further broken down in this intestinal epithelia. Finally, parts get absorbed into, in this case, a blood capillary that's draining into a vein. Over here, I already made mention of this. Here, the fats are getting absorbed into lacteals, which are part of the lymphatic system. Okay, these are tubes of the lymphatic system, and it's a separate part of the vascular system of the body. So this is a great summary of digestion and absorption. I'm going to come back and look at these just a little bit at a time, but largely, guys, this is for you to study on your own. So carbohydrates, let me make a couple comments here. Carbohydrates will include cellulose, which is largely indigestible, and yet it seems to be important for the microflora, the bacteria that exist within our intestines, and perhaps also for mechanical actions in our side of our intestine. I believe right now in modern medicine, it's fair to say that we think a good amount of fiber in your diet, which is what we're talking about, is a very, very healthy thing. So it's good to have that. Another type of carbohydrate is starch. And this is by far, as it says, the most digestible. Starch starts out as oligosaccharides broken to disaccharides and then finally digested to glucose, which is absorbed in the small intestine. Proteins. This is going to be more from meats, but could be egg, for example, and there are some nuts and various seeds that provide good sources of protein just as well. So the amino acids are the breakdown. Those are the monomers of proteins. We get dietary proteins that are being broken down. Interestingly, what also is broken down and absorbed are the enzymes themselves. So digestive enzymes are digesting each other, and those are breaking down. And also then we talked about the epithelial cell turnover in the stomach, said that those cells, maybe every 3 or 4 days are replaced. And so what is lost is digested, and you're digesting part of yourself. And really any of the epithelial cells all throughout the upper digestive tract will be digested farther down in the digestive tract. It's kind of a weird thing that I don't think most people think about. Proteins, here, part of this figure. This is figure 2529, as it says, that precedes, but it's a full page in your textbook that very nicely shows how things are broken down. And then ultimately, after they make their way through the columnar epithelium of the small intestine, get absorbed into the bloodstream. Lipids. Well, lipids, some are digested in part by lipases. And so, as you can see, 10 to 15% of the fats are digested before the chyme even makes its way to the duodenum. So before the small intestine, you are absorbing 10 to 15% of the fats. Before the small intestine. Fats are hydrophobic. I hope you know that. And in that way, then, the enzymes that break them down are largely only working at the surface. And so it's the surface area of these fat globules. And therefore, breaking down fat globules will increase the surface area that's exposed to the enzymes. Okay, so break them down. Break one big fat globule down, so just like a big bowl of jelly. And now you break that bowl of jelly down into other subcomponents, and it's going to have more surface area. So, less than bile salts are also helping to break up these globules in a process that we call emulsification. And the smallest of some of these fats are called minute droplets of bile, called micelles, made in the liver, make their way into the duodenum, and they help to absorb then lipids. And here in the next figure, which is on page 979, really helps to break down in just a gorgeous way digestion of lipids. And notice that their final fate is to go into a lacteal, again, part of the lymphatic system. Here's the little micelles that are in bile. So here's that summary again, 2531, that I really want you to know everything in this. It's just so important. I mean, in the digestive system, we're talking about how we benefit from the things that we eat. And this is such a foundation of you understanding how it is that you support yourself. So this is an enlargement of the top of that figure, so just reaching only the surface of these epithelial cells. And now this is the bottom of that figure, so I can make things a little bit better. And this is the microvilli that are increasing the surface area. You notice that some things are broken down first before they're absorbed by the epithelial cells, and then the epithelial cells may only be transporting the amino acids in, or there may be additional breakdown of whatever's absorbed. Now, that lactose intolerance. So lactose is a milk sugar, right? And typically, until about age 4, humans are producing an enzyme, lactase, that's helping to digest breast milk. But it takes energy to make lactase. And if you think about it, in most natural environments, so think about a dog, think about, wolves or foxes, or a raccoon, or squirrel that are all breastfeeding their babies, the babies grow up and they stop drinking milk. It's the very nature of how mammals grow up. And so this milk as a source of nutrition is pretty much limited to the first early periods of development, after birth. So it's adaptive to actually stop producing this enzyme. So after age 4, most people produce very little. If you don't have lactase or much of it, then what's going to happen if you ingest some milk products, some dairy products? And the answer is the lactose, the milk sugar, is going to pass undigested to the large intestine. Lactose is going to increase the osmolarity of what's in the intestines, and that's going to cause water retention, which leads to diarrhea. There also will be increased gas production because there will be some fermentation of lactose, and thus it can be quite uncomfortable. It turns out the lactose intolerance is not that common in Caucasians or people of mostly European ancestry. So I am not lactose intolerant, so I am lactose tolerant. I love dairy products as an adult. But interestingly, people who have a genetic heritage going back to the continent of Africa historically seem to actually have lots of lactose intolerance. In the Mediterranean region, it's closer to the African situation or condition. And likewise, if we look at Asians, people of Asian descent, continental Asia, that we find that they are very, very lactose intolerant. Okay, lactose tolerance has evolved in adulthood in a number of places, and we think it actually has occurred through different mutations. You can look this up if you'd like to on the internet. It's kind of an interesting story about evolution. If clearly people have cows and those cows produce milk, and that's an important part of the adult diet, we could expect then natural selection would favor those individuals who, as adults, could then utilize this dairy source of nutrition. And so we think that that's where it's evolved. It naturally is the condition to be intolerant, but the tolerance has evolved in a number of places. So we looked at carbohydrates and proteins and lipid absorption. There's also then absorption of nucleic acids, and then vitamins, minerals, and water. Nucleic acids have their own sets of enzymes. Pancreatic juice has enzymes that are breaking down DNA and RNA, and that splits them into the components that you probably learned are the components of nucleic acids. Okay, so there's phosphate, there's ribose or deoxyribose as sugars, and then there are the nitrogenous bases. And there are specific membrane carriers that are selectively bringing in these nutrients. By the way, none of those nutrients here are significant sources of calories. Okay, so what I talked about before, carbohydrates, proteins, and lipids, those are the main sources of calories for us. So those are the macronutrients. These are more the micronutrients. So, vitamins are often absorbed simply with little change. Fat-soluble vitamins tend to be absorbed with other lipids, and so they are ingested without fat-containing foods. They are not absorbed at all, but are passed in the feces. These might be more likely absorbed through simple diffusion, but my question, why taking daily vitamins is best with a meal? This is what we do in our household. We have a set of evening vitamins, a multivitamin that I recommend, and we take those multivitamins with our meals, and we're eating a lot of salads in our household now. So there's salad dressing I use, oils such as olive oil, which is very healthy for you. And so when I take these vitamins, the fat-soluble vitamins are going to help be absorbed by the fact that I took those vitamins while I was eating a meal that had fats in it. Minerals absorbed all along the length of the small intestine, as you can see. And here's some specifics about some of these. We didn't talk much about the chlorine and bicarbonate interactions in the stomach, but this is reversing those reactions in the stomach. So we're not going through many of these mechanisms for these electrolytes. All right. How about water? Well, we consume a lot of water, and we should, because most chemical reactions in our body are occurring within water. So the digestive system is one of the really important systems in our body that are regulating water, of course, because it's where we take water in. Of course, kidneys are helping to regulate the amount of water and helping to get rid of water if we have too much. Water absorption is simply osmosis. And so that means if you have more salts or organic nutrients within the cells, then water will tend to move where they are more concentrated than from an area where they are less concentrated. Large intestine is where we absorb a lot of this water as feces are formed. And if the large intestine does not absorb enough water, then that's going to lead to diarrhea. And this might be simply because somehow the intestines are irritated and feces formation simply passes through too quickly. There's not enough time for the water to be absorbed. And then this is another possibility. There may be a high concentration of solutes there that are keeping the water in the feces themselves. The reverse, of course, is occurring when too much water is absorbed. And this may occur because something slows down the passage. So something that you might have even, it's kind of one of the weirdest ads that I saw, but for people who variously are on opioids, so like morphine and codeine, or who are addicted to heroin, what those drugs do is slow down peristalsis. And what is characteristic of people who take codeine or morphine or are heroin addicts, they have constipation problems. And so there was an ad for a drug that essentially said, if you're on opioids and you have a problem with constipation, you could try this drug. And I don't know if you guys know, but probably before most of you were born, it was a time when we did not advertise pharmaceuticals, prescription-based pharmaceuticals, on television. And that now has become a major source of revenue for television, is all these different prescription medications that you see. But something that is as specific as helping people who are constipated because they're using morphine or other opioid kinds of drugs, that's a pretty specific ad. So I don't know if you've ever seen that, and honestly, it's quite possible that if you saw the ad, you weren't sure what they were talking about. So, what could lead to constipation? Slowing of peristalsis, so that if the feces spends too much time in the large intestine, it gets quite firm and hard and could collect up and be hard to pass. All right, speaking of the large intestine, this is now the end of this chapter 25 discussion. Large intestine receives, depends on what you're eating, and actually has to do with the amount of fiber. So if you've been eating an awful lot of indigestible fiber, plant material, then your large intestine will probably receive larger volumes, and your feces will be greater. And that can be a healthy thing to kind of stretch and move along things that are in your large intestine. So it varies. How much you're defecating can vary a great deal too, but certainly what the large intestine receives and then what is released, what is defecated, should be a great reduction because of this absorbing of the water that's occurring in the large intestine. And of course, elimination of feces is what we mean by defecation. We don't need a photo here. So here's large intestine. We'll talk about sections of it. Large intestine begins at the cecum. Here it says cecum right here, and there's the ileocecal valve. So where the ileum of the small intestine empties into the cecum, that is the ileocecal valve. So it's the start of the large intestine. The appendix is this additional extension right here coming off. You might have heard that the appendix is a vestigial structure. It's an outpocketing here of the gut. The appendix, I believe, has been shown to house lots of bacteria and might be a special place for certain bacteria to lodge or be located. That might be an important part of our health. We're learning more about the microbiome and these kinds of things. But a person can live with the appendix removed. And this is a part of the immune system, and it may be an interaction between our immune system and the microbiome. So in general, here's what the large intestine does. From the cecum, there is a cranial movement of this developing fecal matter in the ascending colon. Then there's a sharp turn to the left, so this is the left side of the person's body. This is called the transverse colon, and it's located right near the stomach. And then the descending colon right down here, the sigmoid, because it's kind of an S shape right here, sigmoid colon, and then the rectum, which is largely involved with fecal matter right as we're getting ready to defecate. So as things accumulate in here, this is that urge to defecate. Sometimes people might think that they have a stomach ache, when in fact it's movement of materials in the transverse colon. If things are moving quickly through your large intestine, you might feel a surge up here on the right, a big gurgle. And then if you feel a flush move from your right to your left side right here, it's a sign that things are moving very quickly. It's likely that your intestines are quite upset. And I would tell you that by the time you feel a surge from right to left, you might have a matter of only 5 minutes or 10 minutes to get yourself to a toilet. So if you're in a shopping mall or you're on a street, you feel this kind of gurgle, you should know that this is where you can use your knowledge of human anat and phys to get yourself to the right place so we don't have a horrible accident, okay? Because if things are moving along that quickly, you may not be able to resist the surge once things are propelled down here through the sigmoid colon and finally into your rectum. So here's a nice 2532 figure going through a lot of this anatomy, and you'll be able to see this in a cat. Here now taking a look at the terminus of the large intestine, and that is the rectum. This is where fecal matter will collect. just prior to defecation, and a little bit about that anatomy. So, you know, this is a part that we're wiping when we're in the toilet. This is a part where we could have problems like hemorrhoids, and there's not much discussion. I think a lot of people don't really understand what's going on in this end of the body. Okay. So the anal canal is about the first 3 centimeters, which is about 1.5 inches inside the anus. So here's the anal canal. Here's the rectal valve. Here's what's called anal columns and the subsequent sinuses. And you can see they're releasing a lot of mucus that's going to allow things to quickly kind of squeeze right out and slide right out. There are 2 anal sphincters. So right here you can see an internal anal sphincter and an external anal sphincter. The internal is smooth muscle and it's involuntary. So once there's fecal matter here and there's a sense or an urgency to defecate, this will on its own relax. You can't control this part. The external anal sphincter might be something that you are working at and pinching in because you don't want to mess your pants up right now and you're trying to make your way to a toilet. There's a phrase that students will use, other people will use, a turtle. So you got a turtle. And what they mean is feces is sort of past the smooth and you're pinching it in and it sort of feels like there's even a protrusion of some fecal matter already starting to come out of you. And that's very likely a consequence of the internal anal sphincter already relaxed. And you were doing all you can to hold things back with the external anal sphincter. But again, if this is diarrhea, good luck holding that back. It's very likely not going to be successful. Here's some very large veins that are in the area. And these veins, through various motions that people may have, so straining sometimes in the toilet or sitting too much in a chair, can lead to the enlargement of these veins, and they're called hemorrhoids. Hemorrhoids are this permanent distension of these veins, so they remain inflated and they can intrude into the anal canal. They may be even so big that they are extending out of the anal canal. And if they rupture, it could cause a lot of bleeding. So I had a student once who had very large hemorrhoids and they ruptured and he had to go and have surgery quickly. So this can be a very serious thing. It's also can be very painful. So hemorrhoids is a thing you might joke and chuckle about, but boy, if you have them and they're a problem for you, if they become a problem enough, a surgery may be necessary. So here's just an up close of exactly what we were talking about. I guess also in this figure, now that I got it right here, anal intercourse is where probably a penis is inserted into the anus, and there's at this portion, the very end portion of the rectum, so in the anal region, there are cells that are very much like the inside of your mouth and like the inside of the vagina. It's stratified squamous, non-keratinized epithelium that's good at resisting friction, and it's also better if it's wet. So we talked about mucus being secreted in this area. But that's only about the first 1.5 inches or so. And so if a penis is thrust or anything's thrust much beyond, that about 1.5 inches, so we'll say even 2-inch area, then something is being shoved into the rectum. And the rectum does not have the stratified squamous, non-keratinized epithelium. Instead, it is more easily torn. And if it is, then blood could be leaking into the rectum. And if a person were ejaculating into the rectum, then whatever's in the semen would mix with the blood. And this was the major problem in the transmission of the AIDS virus, of HIV, because in a number of gay men, they were having anal intercourse. And so someone who was infected If they were the one inserting the penis, then they would deposit semen with HIV, and it was very easily exposed to a blood supply here. I don't know whenever you're going to get this explanation, and so it's anatomy and it's a public health issue, and we're focused on COVID now. We've made so much progress with HIV and AIDS, but this was a transmission problem. Here's showing you just some of these cadaver highlights and the various parts. You can freeze this and enlarge it if you'd like. Here using x-rays, so an imaging. And this may where they have some kind of material that's opaque to an x-ray. And so you swallow something, and at a certain point they can then take some imaging and take a look at some real detail of the outpocketings of the intestine. And you should know that having a colonoscopy, so they're going to insert usually a flexible tool up into your colon, and they're going to look for the beginnings of tumors. And so hopefully, if they find any, that they're tumors that have stayed put and they can nip them off. And having a colonoscopy starting at about age 50 is recommended. And now they've lowered that age if you have any risk factors you might want to look into. Now, I know you're far from that right now. This is done under anesthesia. I'm 64 years old right now, so I had one at the age 50 and I had one at the age 60. It was done at the Litchfield Hospital. A person who does many of these, and they knock you out. And as far as I know, I was knocked out. I was on a stretcher. They wheel me into a room, and I count to about 3. I'm knocked out, and then I wake up. So it's nothing to really be too worried about. The worst part of it is you have to flush your intestinal system by drinking a pretty horrible liquid that's very, very thick. And you have to work to make sure you don't gag and throw it up the day before. So the day before, you're pushing this stuff through you, and it tries to empty out your intestines, so that the physician who then does the colonoscopy can very easily inspect this area. And of course, it's a scope with lighting. They'll also inject, by the way, some air into your colon to kind of inflate it so that things can be stretched and we can more easily diagnose problems. Here's looking at the different parts of the rectum and the anus, the anal canal here and the rectum, the external anal sphincters that we talked about, the internal anal sphincter much farther up and smaller, and over here the anal column. I don't think we talked about the transverse rectal fold. This is the histology of the large intestine. Here's the mucosa, and my goodness, there are so many mucus cells, lots of mucus cells here. The goblet cells here are in this bluish coloration, and that's most of what you're seeing. That's one thing that I look at when I see something on the intestine right away, and I think, oh my goodness, look at all those goblet cells, and that must be, I think, a large intestine. Notice that the surface of the mucosa is fairly flat. Where did we see this before? And the answer is the stomach. But the stomach did not have a bunch of goblet cells. So when I see a mucosa like this, it's clearly part of the digestive tract, a flat surface. I know it's not small intestine. Right away, quick flashes in my head that if we got these columnar cells, that this must either be stomach or large intestine. And then as soon as I look here, we do not have, as we would in the stomach, these glands that come down and fork. Instead, we just have what looks like, I would describe this as clefts, as if this was, I don't know, bread or cheese, and you used a knife to make cuts coming straight down. That's what I see when I look at this epithelium. So anal canal, non-keratinized stratified squamous epithelium, no circle folds or villi, and there's a lot of lymphatic tissue in the submucosa. We'll look more at this when we do the histology in lab. I said the microbiome has been much more appreciated in the last decade, and it's still a very hot area. It looks like there are some diseases that might be addressed by introducing other microbes into the intestine. We know that when you take antibiotics, it is very disruptive of the microbiota, and it may favor one type of microbe over another. It creates a tremendous imbalance, and that has to be restored, or you could be suffering from diarrhea or other problems. If you see right here, we know of at least 800 species of bacteria in the large intestine. Very diverse. I was listening to something like a TED Talk from a person who is an expert in the microbiome, and they said something it's very interesting. It's a challenge to you, and it's a challenge we've taken up in our family as well. Now, the woman said, look, if you want to support this healthy diversity of bacteria in your intestines, then you need to eat a lot of diverse things. And her challenge was, every week, can you eat 30 different types of plant material? Okay. So wheat and corn would count as 2. There are many different types of lettuce. So in our family, we have a salad most nights. A major portion of our dinner is a large salad. And we buy it like you get at Walmart, the spring mix, which could have up to 5, 6, or 7 different types of salad lettuce of some sort, like romaine and spinach and other things. And when this woman was talking about this diversity, she included any other plant material that you might be adding to a salad. So for us, carrots, we had sunflower seeds. That's yet something else. We have soybean, because we use the fake Bako bits that are actually made of soybean. I put 2 different types, sometimes 3 different types of olives on there. I also put on peppers, so pepperoncini peppers. I often put mushrooms, so mushrooms also count. So plants and mushrooms, I should say, and fungi. And you just go through the day. Potatoes, of course, is another category. Any other kind of vegetable, so green beans, any other kinds of beans that you eat. In a week, try to eat 30 different types of plants and or mushrooms, right? Just as well. It's a big challenge, and you can include spices. So if you got garlic, that counts. And the point is, when you're eating this way, diversity is good. Diversity in the diet is good because it covers your macronutrients and your micronutrients. And diversity in your diet is good for the diverse microbes that live within you. So, in biology, the one thing that we have been promoting as long as I've been around and in the field is diversity. Diversity is good, good, good. Diversity gives you options. It promotes diversity. Diversity promotes diversity. Diversity in the diet promotes diversity in the microbiome. Diversity in a forest promotes diversity of the critters that live in the forest. We know that diversity in humanity provides some of the same benefits, which is when you have diverse experiences and diverse backgrounds and diverse perspectives, you're better able to understand and talk through problems. Because if you're all thinking the same way, then you're not likely to come up with new and different approaches. So I know and I support the recent emphasis on diversity in our campus and across the country, but it's something that biologists have long supported. It's just, when I hear diversity, I just think strength. I think that it's the resiliency, it's the ability to address issues. It's just fundamentally good. Okay, I kind of have gone on a little while there, but diversity in the diet supports diversity in the intestine. So you want to stay tuned, and it's really, really a good subject to investigate. What do these bacteria do? Oh my gosh, the list is very long, so here's just a little. They're helping to digest some things that are otherwise undigestible, like certain kinds of carbohydrates and cellulose. They also synthesize some very important vitamins. The list is very, very long. Okay, I promised a little bit about how it is that flatulence is formed and what's going on there. We're talking about farting here. A person produces, as you can see, about half a liter or 500 mLs of gas a day. Most of it is reabsorbed in the large intestine. Some could be that you've swallowed air. Here I am talking endlessly on these lectures, and I'm wondering how much air I'm swallowing in this process as I'm swallowing my saliva or breathing in. But certainly, some of the chemical processes that bacteria engage in will generate some gases. And some of them can be really bad smelling. So you may know that there are certain foods that you can eat that lead to some very, very bad smelling gas. And people joke about this all the time and laugh about it, but this is the thing. There are certain things that you can eat that can lead then to these very bad smells, and the sulfur kinds of smells tend to be some of the worst. Hydrogen gas can even be produced, and it turns out that surgeons need to be aware of this, because if we're doing some kind of electrical cauterization where we've got some bleeding and we're going to kind of burn the area and stop, well, if we did that, there could actually be an explosion of the hydrogen gas. So they might want to flush the area, and I don't know the procedures for this, but it's a thing. The passage rate. So how long does it take from when you ingest something to when you defecate can be very, very variable. Okay, V-E-R-Y, very variable. You might notice, for example, that if you've eaten corn or garden peas or something else that's rather undigestible, you know when you ate it if it was just one meal of it, and then you can tell when you defecate it out, so when it arrives into the toilet bowl, right? Passage rates can be a matter of less than half a day, so it could be less than 12 hours. And of course, in some very slow passage rates, it could be 48. But here's an important thing to know. A meal does not progress through your gut together. Some things move faster through your digestive tract than others. So, water might move quickly through your digestive tract while other components get slowed down. So what might feces consist of? Lots of bacteria, undigested fiber, fats, and all these other kinds of things that come out of you. But what I think is kind of interesting here is that I probably, years ago, was thinking that, yes, a meal, so if I eat one meal and the 3 or 4 things that I might have had for that meal, I'm thinking that they're all coming out of me at the other end. I'm defecating them all the same, and it's not. You should know that they don't all move at the same speed, okay? Now, the urge to defecate, and that's what we're going to talk about here at the very, very end. Materials have moved through. They've made their way into the rectum. And what has to happen, that we said that inner smooth muscle, sphincter, is going to relax on its own, but the external has to be relaxed as well. What we tend to do when we do abdominal contractions actively and try to push it out is that we essentially try to exhale but not let the air out. So you hold your breath like you're holding your breath, right? So you could pinch your nose and close your mouth. You don't have to pinch your nose. And yet you create an internal thoracic pressure like you're trying to breathe out. And the effect is to push your diaphragm downward, and as it pushes downward, it compresses this abdominal region. And as it does, then it tends to force out the material, the abdominal material. So that's kind of the grunting or the moaning or pressure stuff. It helps maybe to lean over and compress your belly. You could do this also by lifting up your legs. All of that creates pressure that helps you defecate. And by the way, these same maneuvers help you if you're giving birth, so obviously for women. This generation of this internal abdominal pressure by essentially trying to breathe out but not letting the air out is called the Valsalva maneuver. I don't quite know why it needed a fancy name, but it's using your thoracic region to generate abdominal pressure. Here in figure 2533, they're going through a little bit of the reflexes that are involved. So the rectum is filling. So feces is in the rectum. It stimulates stretch receptors. That will get your attention. Signals then those stretch receptors to the spinal cord. And then there's some parasympathetic fibers that are going to cause this internal anal sphincter to relax. Some other signals that are going out that are controlling the voluntary part. And defecation is, unless it's diarrhea, is probably going to require then that you relax these external muscles. But, things can happen where you can't even stop that. The Valsalva maneuver might help you then to generate the pressure, but in some cases the feces aren't very big. If they're firm, if things are well lubricated, you don't have to generate much pressure to defecate. And this is the logical way, it seems, to end the chapter. So we have followed through from initial ingestion, taking something into your oral cavity, and looking at mechanical and chemical digestion all throughout the process. As we said, something that a bite of apple that is never digested technically has never entered your systems of your body, but I would argue it was in your body because it was inside your body, but it wasn't ever absorbed and passes through you. So here's where we're at in the course. We've just finished chapter 25, lectures about the digestive system. That's the only chapter on lecture exam 1. Okay? And that lecture exam is on February 4th during the lecture period. Okay? We will do that in person in the Zoe Lab. The next lecture exam, and there are a lot of them, guys, involve 2 different chapters, 26 and 22. I just mentioned to you at the start of this lecture that 26 is taking a look at nutrition and metabolism. It's only these 3 parts of that chapter, and very specifically on the testing recall and all the word roots and true false. This is off the syllabus. This is what you do on your own, and this is where the next lecture, so the last lecture for this week, for I think the 28th of January, will start the respiratory system. We have lots to address in this semester, so I got to keep going. There's no time for a lot of time out. But so far, we're doing fine. It's good to be on track like this. So if anything, we're maybe a couple lectures ahead. So that's it. This ends then our look at chapter 25. This is a little longer of a lecture, but I thought it best to finish this up and end it right here. All right, that's it. Bye-bye.